Hello everyone. This video is to explain the working and functioning of laminar airflow. So laminar airflow happens to be one of the most important instrument or equipment in a microbiology laboratory wherein we carry out almost all microbiological experiments, starting from inoculation of microorganisms to staining to plating and so on. So herein, first of all, we need to understand what are different components of a laminar airflow. So if we look on top of the inside the hood, if we see, we have a HEPA filter towards the inside. It is called as HEPA filter, high efficiency particulate air filter, that does not allow microorganisms to come and contaminate the plates. So the purpose of carrying out experiment inside a laminar airflow or a biosafety cabinet is to ensure that microorganisms do not contaminate other substances. Also, the microorganisms that we are trying to isolate also do not get contaminated by other forms of microorganisms. So to ensure that, at the beginning we need to sterilize the entire hood. So for that we have a tube. If we look on top here, we have an UV tube and also one tube that is working tube, that is a normal tube that helps us during functioning, and this is the UV tube that actually is switched on before starting the work inside the laminar hood, and this UV radiation actually kills all the microorganisms present inside the hood. And there will be a blower. So normally how it functions is, keep it closed and then we switch on the UV. So if you can see now, inside there the UV light is on, and as a result of that you can see the light switched on and all microorganisms present inside will get killed. So we'll leave it on for a certain time, and then after the sterilization process is done, then we switch it off, and then we switch on the working light inside. The fluorescent light is on now, and along with that we switch on the blower. So when we switch on the blower, then any particle that is present inside will get blown out. So there will be a laminar flow of air inside. That is why it is called as laminar airflow. And after that we open the hood and then we light the Bunsen burner, because whenever we work with microorganisms we should do it near flame near fire, so that nothing is contaminated and we get pure isolated colonies. And before that, before starting with our isolation and inoculation, we will have to clean the surface with the help of alcohol, and after that we can carry out our function. So that is all about the functioning of laminar airflow. After we are done with our work, we again close it down, and if there are microorganisms, if there is a possibility of microorganisms being present over there, we can again switch off the blower and fluorescent light, and we can switch on the UV once again. Say we will switch off the blower, switch off the fluorescent light, and switch on the UV again, so that all microorganisms that might have been present after we have carried out our work over there will get killed, and then at the end we will switch off the UV and the job in the laminar airflow is done. So that's all about the functioning of laminar airflow.